Well, hello class, this is Fabiology DNA Transcription. So our goals for today, by the end of the video, students will be able to explain the differences or compare and contrast between DNA and RNA and explain the process of transcription. So you'll know you mastered this if you're able to draw a picture and explain that picture to somebody else and also perform transcription from a strand of DNA. So any sort of template strand I give you, can you make mRNA out of that? So here we go, transcription. So what I suggest is that you pause the video and make sure that you have some different colors, whether it's pens, crayons, colored pencils, because you're going to want to do some drawing. So as we're doing this video, pause it when you need to, okay? Pause and take notes. So here we go. Let's start with the central dogma of molecular biology. So it sounds really big and complex, but dogma refers to the rules, right? We have a dogma for different religions out there, so now we have our dogma or our rules for molecular biology. So here's the rules. We have DNA, so we can have our DNA strand. So if you're taking notes, I suggest you scribble some little lines there, label its DNA. Then we have transcription happen, and we have mRNA. So what do you notice is the first difference between DNA and RNA? The first difference really is that it's single-stranded. So instead of having two lines, we have one. So instead of being double-stranded for DNA, so D for double-stranded, D for deoxy, that's kind of how I remember it. Once we transcribe it, we're going to make it into what's called mRNA, and that's single-stranded. The last thing is that we're going to translate it, and this is what we'll get to in another video, is we're going to translate that code into something that the ribosome can actually read. So this is where you want to think about languages. When we're talking about language, when you transcribe the text, you want to write it so that somebody else could read it. So let's say you speak white Hmong, if we're talking about the Hmong language, because there's two dialects, white and green. The nucleus speaks white Hmong, but the ribosome speaks green Hmong. So even though the similar words and it's almost identical, there's always those one or two differences that we don't quite know. And so this is where you have to think of your language arts teacher or your foreign language teacher. They're telling you something that you're going to transcribe so you can explain it to me who doesn't speak that language. So this is what we already know, and this is what we've talked about before. So genes are made up of DNA sequences. So thinking about any genetic disorders that you've researched, thinking about the last unit of how genes and Mendel was working, this gene code, ATGGCAT, is going to be different in a completely different gene than CCGGTAC. So even though the letters are slightly the same, one letter different or a few letters different causes a completely different gene to be made. But ultimately, if we think back to our genes and think back to Mendel, the phenotypes that we see are really RNA, because the DNA just codes and the RNA then will make it into a protein. So the RNA goes to our ribosome, our organelle for protein synthesis, and will be expressed there. So it's really the RNA that we're seeing, because the DNA codes for all those proteins. So it's like the blueprint of the DNA and then the message from the RNA. So here's what we have. Boom. We have RNA, which is ribonucleic acid. So we already talked about one difference between DNA and RNA when we're talking about the single strands of RNA. We're also looking at ribosomes, the ribose sugar itself. The sugar has a similar shape and structure, except it's not deoxygenated. It's a ribose sugar, so it has one more oxygen molecule on there. Still has our balloon, house, and mailbox setup, so we have our normal nucleotide setup. But the big thing that we can see here is a base pair change, and that's one of the most important things to understand from this. So if it's me taking notes, I would definitely highlight that you have a brand new letter. The base pairs for RNA are A, G, C, and U, which is uracil. So there's no more thymine. So as we're copying our DNA strand, we're having to put uracil instead of thymine. And that's that idea of different languages. It's the one word that is not the same. We don't speak the same language. The nucleus and the ribosome, completely different languages. So here's this transcription. Boom, the official definition. We could go and make a very big and boisterous one of the process of blah, 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 right? We don't want to do that. Let's make it simple on ourselves. It's making mRNA from DNA. Transcription is basically making RNA from DNA. So this is where you're going to want to pause and draw this picture. Notice the different colors. The red and blue is all DNA, and that red and blue is to show us that idea of semi-conservative, which is what we learned about DNA replication. So it doesn't really matter. You could keep it the same if you want to keep this idea of it's just DNA and we're focusing on DNA versus RNA. But in the end, you want to look at those color schemes, because the green one is our new code. It's our mRNA code. And you can see already that we have AUG, or our start codon, as we go along. The other thing we want to notice is the stuff that opens it up, this giant circle around it. That's RNA polymerase. This is nice about RNA and protein synthesis, is because instead of having 3 enzymes that we do for DNA replication, we have one. RNA polymerase. It's an enzyme that opens it up. It'll close it after typing the code. So open, type the code, close it back up, and send that mRNA outside the nucleus to a ribosome floating out in the cytoplasm. You can also see between T and A and C and G, we have these bonding pairs there. Those are our hydrogen bonds. So we can see the same setup. So if we're comparing and contrasting, we can still see similar base pairs. Again, it's just that one letter and that single side DNA template. So again, honestly, pause and draw this in your notes. So like I said, there's 3 steps. DNA opens up, mRNA is made from DNA, and the mRNA leaves and the DNA closes back up. And that's all from one enzyme, RNA polymerase. So open, type the code, close it back up, send it to the nucleus or to the ribosome. Why do I keep saying mRNA? Well, there's a reason. There's actually 3 we're going to learn about. First one's messenger RNA. This is the one that is copied from DNA, and it has what's called codons. So codons, think of the code. Message, it's the letters, it's what we're mailing to somebody, okay? And this goes from the nucleus to the cytoplasm. A codon is a set of 3 nucleotides, or 3 bases of mRNA. Each one is for a specific amino acid. So an example, if this is our mRNA strand, every 3 letters is a codon. And that's important because when we get to translation, we're going to have to look at codons. And so what I like to do when I see my mRNA strand is I like to just draw straight lines every 3 letters. It keeps me on track, and it keeps me knowing which codon's which. So here we go. This is our DNA strand, and we want to go and make this into RNA. So we know we're going 5' to 3', and we have now A is going to bind to U. So instead of T, it's a U. So we have U, C to G, so U, G, U, G, A, C, U, C, A, U. And we're done. That's transcription. That's it. If you can do that, if you can replace all the U's and the T's, you're all set. But it's not that easy, because before you even send it somewhere, you have to edit it, right? It's a letter. You have to proofread your letter so it doesn't have any mistakes. So after you copy it, you edit the mRNA strand, and then it goes into the cytoplasm. So what are we editing? This. And this is something you might want to draw as well. Within our DNA strand, sometimes there's just this junk that doesn't code for anything. Those are called introns. Introns need to be cut out, otherwise our protein is going to be made poorly. So we cut out the introns, and you can see the lighter colored blue there. That's the intron from the DNA code. And we have the other one, which are exons. Exons are what we keep. They actually code for something. So after we make our pre-mRNA, or we make an initial copy. We're going to cut out the introns and put them all together. And that's it. Then we have our final mRNA that can leave the nucleus and go into the cytoplasm. So here's some practice problems for you just to kind of process these thoughts. Again, I encourage you to go back and rewatch parts of the video, make your drawings really nice and clear. But we have a DNA strand. I want you to turn that into mRNA, and then think about if codons 2 and 4 are introns, what will be the final mRNA strand? So go back, put those lines in, or number the codons, right? The first 3 letters is 1 codon, the next 3 letters is 2, and so on and so on. What do we do with introns? What's our final mRNA going to look like? Thanks for watching, and have a good day. Bye.